Hi everyone, thank you so much for tuning in. Good evening to all of you, and we're going to be continuing the discussion on the immune system, part of the immunology exam in my microbiology class. And so I'm going to just split this up. Actually, there's two parts to adaptive immunity, so I'll just go through the first part here and then we'll pick up the second half once we get there. So adaptive immunity is different from innate immunity because innate immunity is kind of like the generalized response. You have inflammation, you have fever. You're only looking at pattern recognition in innate immunity. However, in adaptive immunity, this is when you get your specificity with antigens and antibodies, and then also your lymphocytes. Right, so a little bit of history. So in the 1800s, we had huge cases of diphtheria, which is a toxin, and the scientist named von Behring, what he did is he took the diphtheria toxin and he puts it into guinea pigs. And these guinea pigs actually recovered from diphtheria previously. What happened was they were again exposed to the toxin. They did not get ill. So something in these guinea pigs that already had the toxin must have been killing any newly introduced forms of it. So they must have had something like an antitoxin. When he mixes toxin with a serum from a guinea pig that had survived and injected into another guinea pig, it remained well too. So if we had a second guinea pig get injected, this one was okay as well when we make that serum with the first guinea pig. And then, so in Berlin in 1891, the first antitoxin was given to a kid and they recovered. So he wins the Nobel Prize in Medicine in 1901. So a little bit of a recognition from the body. There's an antitoxin maybe involved, and then whenever you introduce that same toxin or same pathogen again, the body's able to fight it off. Alright, so let's get into the adaptive immunity system. And our first stop will be the types of lymphocytes. Again, lymphocytes are the white blood cells in a category of their own. These are going to be still leukocytes, but think of them as like a subcategory. We talked about 3 categories of leukocytes yesterday, and we had the granulocytes. We also had the mononuclear phagocytes, and then now we're going to talk about the lymphocytes. So, in lymphocytes, we're going to have 2 categories. We're going to have immature lymphocytes, and these do not have the fully developed antigen-specific receptors. Keyword there is don't. This is different to the naive lymphocyte. Actually, there are 3 categories. Sorry about that. We also have the naive lymphocyte. I guess I was a little bit naive there, giving the wrong number of categories, but we actually have 3. This is the second kind. A naive lymphocyte has never encountered antigen. So AG, never encountered the antigen, never seen it before. When we get to our activated lymphocytes, when we say activated, this means it has received the specific signals. And then, so when that happens, we get proliferation, so numbers will increase. And when the numbers increase, they'll make effectors as well as memory lymphocytes. Effectors are going to be short-lived and they're going to be the primary response. However, the memory lymphocytes are going to stick around. These are going to, of course, then be the secondary response. Again, lymphocytes are just another category of white blood cells. Okay, so now let's talk about their receptors. So here we're going to be talking about how they get that specificity down. Right. So B cells and T cells, again, are two types. We also have natural killer cells, or NK cells, but right now we're going to talk about B and T cells a little bit. And these have membrane-bound receptors. So they're going to recognize specific antigens. So AGs. B cell receptors. So in the BCR, a B cell receptor, this is specific to an antibody. The cell is programmed to make. And this is going to be, the receptor isn't the antibody itself, it's just Y-shaped, kind of like an antibody. It's not the antibody itself, it's just a receptor. And then so when we have binding, this is going to create a response. This is going to trigger that response. In the T cell receptor, or the TCR, these are a little bit different. They do not recognize free-floating antigens. Instead, you're going to have two polypeptide chains, and it's going to look like one arm of the BCR. And these, we're going to just have kind of a stick like this, and then there's also something called a CD marker. That's not very important, it's just the structure of it. And what happens with the TCR that is different from the B cell receptor, these must be presented on the surface of another cell. It's major histocompatibility complex. So we're going to look for these TCRs on the surface of another cell, and then this is another acronym. This is the major histocompatibility complex, or the MHC. And then binding will activate it. So on this TCR receptor, or T cell, TC. I'm saying it like ATM machine, you know. So we have this other cell here, and the cell will have an antigen here. This is the antigen. I'm gonna just put AG. It's a circular antigen like that. Okay, so we have a binding site in here, and then this is actually just going to move towards that, and that's how you get your response. On the BCR, you'll have the antigen actually float in. So if this is my antigen here, this is just going to go and attach to the receptor, and that's telling the cell to start making that specific antibody the cell is programmed to make. Okay, let's move on to the lymphatic system. Of course, lymphocytes, lymphatic system. There's a connection here. In the lymphatic system, we have a collection of tissues and organs. These are going to bring the B and T cells in contact with the antigens. They need to get to those areas. And then in this system, lymphocytes will encounter the appropriate antigen. We also have lymphatic vessels. So lymphatic vessels. And these are going to be fluids, and then they're going to be leaving the body's circulatory system. And then not all the fluid will return to the capillaries. And then the rest of it, goes to the lymphatic tissues and organs. Therefore, the lymph actually empties into the circulatory. So you'll have some of these tissues, and then, near the capillary, the excess tissue fluid will become lymph, and it's going to go to the lymphatic vessels. We also have secondary lymphatic or lymphoid organs. It's taking a bit of time, but we're going to try and get through everything. This is where contact with antigens happens. And these will be, of course, the nodes, the spleen, and the tonsils. The Pierce patches will allow for the sampling of intestinal contents. And how they're going to sample the intestine are the usage of M cells as well as the dendritic cells. We're going to also have something called the mucosa associated lymphoid tissue, or MALT. That's again one of our abbreviations. So this is going to actually stop microbes by the mucous membranes. Alright, lymphoid tissues under skin are going to be known as skin-associated lymph tissues, or SALT. These are, again, under the skin. Another acronym. Alright. Let's talk about adaptive immunity again. So we're going to go up here, and again, we're going to be a lot more effective. Let's just use the same color as we were using earlier. We're going to be much more effective because we're going to need that time, a week or more, to build. However, we're going to be very specific in our response. And while we're trying to build it up, we're going to actually be using innate immunity to handle things. Adaptive immunity has both memory and specificity, as we discussed. Memory is this strong response to re-exposure. That's why vaccines are so important to get, because they allow you to use that adaptive memory, which is extremely powerful on the B and T memory cells as well, to prevent re-exposure. The body must distinguish between your own cells and the dangerous non-self. because you don't want your immune system to be going after your own cells. That's a lot of these fortunate diseases that cause that, and that's just not really good at all. Versus the danger. All right. Primary response be our first stop here. Remember, we were talking about the primary response up here as well. The effectors are going to be lymphocytes in the primary response. They're short term, and they're just going to be like the first wave towards antigen first. So we're going to have the outcome of remembering the antigen. And when it's re-encountered, like after a vaccine or during the effects of a vaccination, the secondary response will be much stronger. There are two strategies. In this video, I'll be talking about the humoral, and then in the second half, we'll be talking about the cell-mediated immunity, or CMI. There isn't really a fancy acronym for humoral immunity. In humoral immunities are going to be extracellular antigens. So bacteria, toxins in bloodstream or tissue fluids, and we're going to be using antibodies here. In the cell-mediated, these are going to be dealing with antigens inside a host cell. So, such as when a virus is infecting a cell. Alright, once again, we're going to be focusing on the humoral side of things in this video. Big picture of humoral immunity, we're going to have the B cells, the lymphocytes. B lymphocytes are going to be responsible. These are going to be found or made in the bone marrow. They're going to be triggered to multiply. And then this is caused by extracellular antigens. Again, humoral is about extracellular antigens. There's something in your blood, a bacteria or a viral particle in your blood, and that triggers this response. Afterwards, they become plasma cells. It's like the second step. So actually we could do first, second, and then third afterwards. And then the plasma cells make antibodies. Only the specific antibodies that are triggered by the antigens that are specific to them. Again, specificity is very big here. Of course, the BCR is going to be the membrane-bound version of the B-cell specific antibody. Which we discussed earlier. And the antigen binds, and then this causes a response. This kind of a copy of what we said earlier in this video. And we also need to get confirmation from a T cell. This is what my professor purposely called the checks and balances system. Because again, the immune system is very powerful. The responses, especially from the this adaptive immunity. So with great power comes great responsibility. The antibodies will be again used for protection. And then how they're going to be protecting. They bind to antigen. And then that prevents attachment. Again, a lot of these disease-causing particles or organisms, they need to attach to the cell in order to cause an effect. So if we bind the antigen to an antibody, this is going to prevent that. We could also indirectly prevent or protect using antibodies by tagging the antigen for removal. Two ways, so binding to the antigen itself or just tagging it. The antigen could actually be looked at as the generator of the antibody. That's where we get antigen. So it generates a specific antibody when it's present. And so it reacts specifically with an antibody, a B-cell receptor, a T-cell receptor. So BCR, TCR, or antibody. So when it causes an immune response, this is called an immunogen. So not all antigens are immunogens, but all immunogens are antigens. There are two types of antigen, so usually there are T-dependent antigens. It's our first category of them. And this is where most of them are going to fall into. The B cell needs the T helper cell to be activated. And then we also have, of course, T-independent. These ones are going to activate without TH. Specifically, an example would be the lipopolysaccharide found in gram-negative bacteria outer membranes and the molecules with repeating subunits. Specifically, carbs. These are going to be enough of a red flag for the adaptive immunity system and the B cells to go, hey, we don't need the T cells' help in this. We know that there's something up because we're gram-negatives doing this area of the body. Antigens are going to have a different response in strength varying on their type. So a strong response will come from proteins. A weaker response will come from lipids and small molecules. No response. Usually these aren't immunogens. Epitopes are the small regions of an antigen that are recognized by antibodies. And these could be regions of macromolecules. So if we have this big bacterial cell here, and we have these epitomes all over it, these epitopes. So when we have the antibodies introduced in the system, like so, they're only going to attach to that epitome that they're specific to. Alright, let's talk about the structure and the properties of the antibodies. Usually we'll see them as Y shapes, but there are actually a lot of different interesting forms they could take as well. So, let me just draw a big, giant Y. There's not going to be much to write except for like labeling this. And I'm going to do my best to describe it because I know I'm just going to be listening to this video to study in like a walk or something like that. When I can't actually see what's being written. Alright, let's do this. So, we have antibodies, also known as immunoglobulins. They are usually Y-shaped with two parts. We have the variable Fab region, which is kind of like the V and the Y. This is called the Fab region, and this is going to be variable. This is for the specificity. The Fab region is specificity, so it's very fabulous. The antigen binding site attaches to a specific epitope. So we can have that E again for epitope. It's going to go into that binding site. The fit is precise but reversible, so numerous non-covalent bonds are at the binding site in the Fab region, which is variable. The constant region, which is also known as the FC region, will be the straight section of the Y going downwards. It's where the branches kind of converge in a Y. So this region is the FC, known as the constant region. And this is going to allow the immune system to components to recognize the diverse antibody molecules. This is like their identification chip, basically. And we have two heavy chains and two light chains. Amino acids will fold into characteristic domains, and the disulfide bonds are going to be joining the heavy light chains in two halves. So we have a disulfide here. Oh, shoot, I didn't mean to do that. We're going to have the disulfide bond in between the heavy and the light chains and two halves. So disulfide here, going to be splitting this thing in half. I just draw that quickly. There we go. And again, disulfide bonds to bring these two together. So we have the heavy chain and the light chain. So heavy, let me make this a little bit, I don't know, that's fine. Heavy look like that. However, light chain is only this section here. The heavy chain is like the one half of the Y's branch and then going straight down, while the light chain is only the branch of the Y, if that makes sense. So light and then heavy. Alright, functions of antibodies. There are actually 6 of these, so we're just going to list them. There's neutralization. And this is going to prevent toxins or viruses from binding to the cells. Again, these need to be bound to the cells in order to actually affect them. So if we have this virus here, and we're going to surround it by these antibodies that recognize those specific epitopes on its surface, that virus or that toxin now cannot bind to anything once it's surrounded by all those antibodies. The second function of an antibody will be opsonization, which we've seen in innate immunity when we get to that C3b from the complement system, we're going to add sauce again to bacterium or a particle, and then a phagocyte. That's going to really get it excited. So add basically a marking or a sauce to attract a phagocyte. So if we have the bacterium here and we put those opsonins around it, but in this case we're going to have the antibodies act as opsonins, and that's going to attract this big old phagocyte, and it's going to eat the bacteria with even more energy. We also could activate the complement system. The classical pathway, actually. Remember, we have 3 pathways. We have the leptin, which is going to be finding those mannoses on the bacteria. We also have the alternative pathway where C3b is just always present, since C3, that protein, is just so unstable, so we already have some. The classical pathway, however, we're going to find the bacterium, and then there's going to be those antibodies on it because they're recognizing those epitopes. And then we have our complement protein here, and this is going to cause opsonization by C3b, and then inflammatory response, which is again localized inflammatory, is called containment. And then we're also going to have the lysis of the foreign cells. So this is going to again become C3b, and we kind of talked about what that does. It just causes number 2 up here, opsonization. Alright, the 4th thing that antibodies could do would be the immobilization of the bacteria. So when we immobilize bacteria, we're going to stop their flagella. We're going to block the flagella from being able to be used. We're going to put some kind of like a rubber tire, a rubber boot on the car that's suspicious, so it can't zoom off or trying to identify it. We could also cause aggregation with antibodies, and the 2 arms of antigen bind separate antigens. So the 2 arms of the antibody, I think there's a typo here in my professor's notes, no biggie, I think we got the message. It's just the 2 arms of the antibody bind to other antigens. So it kind of makes like this chain. So if we have a bunch of bacteria here and they're kind of suspicious, the 2 arms of this antibody are going to lock on, and then another bacteria there. And then we also have the antibody dependent cellular cytotoxicity. That is a mouthful. So antibody dependent cellular toxicity. I spelled that wrong. Oh well, I just cursived it, I guess. I don't know. Okay, so this is going to target the cell for natural killer cells. These are going to be going to an infected self cell. So host, infected, and then we have the antibodies around there marking this infected host cell, and then we have a natural killer cell with all those enzymes, acids, whatnot, and it's just gonna really blast that host there, the host cell that's been infected. We have 5 major classes of antibody. I'm just gonna list them out. So we have IgM. This is going to be a pentamer antibody, so it's going to look kind of like a pentagon with all those like crazy Y's coming off of it. This is the first type made, and then this will lead to the complement system when binding happens. We also have the IgG, And this is going to be our regular Y one. It's a monomer. This is most seen in tissues. This also leads to the complement system. We also have the dimer IgA, and this is going to again be found in the saliva, tears, and mucus. It's weird, this one is also the most abundant class produced. Then we have the IgD, which is also going to be a monomer one. This is going to be the maturation of antibody response, so it develops the antibody response. And then IgE, another monomer, so just our regular Y. This is going to be binding via Fc. So Fc, again, it's the bottom straight branch of the Y where those two branches meet. And this is going to be bound to the mast cell and the basophiles. And then the IgE is going to be allergic reaction. Okay, so we have Ngade, M-G-A-D-E, as a fun acronym used by my professor to remember all of them. So M-G-A-D-E, Ngade. And then G for tissues, A for saliva, tears, and most. A got the highest scores, the highest number. D is development. E is the Fc to the mast cell, so allergic, so a chewy kind of, I guess, allergic reaction. Oh well. Okay. Clonal selection. Okay, so when we have clonal selection, this does not have one of those icons in it, but we could still talk about it. This is the process where only cells with a specific, that are specific to an antigen proliferate. This has to deal with, again, specificity, as well as you don't want to waste any resources if it's not the cell that will match with that specificity. So there are actually billions of Bs and T cells. And this means the body could recognize basically a limitless variety of antigens. However, each cell only recognizes one epitope. So therefore, lymphocytes residing in the secondary lymphoid organs wait for a dream antigen. And then in most cases, the B and T cells need confirmation from other types of cells. So that means if we have the antigen X, but we have all these other B cells that are naive, they haven't encountered it. So let's say antigen X is here. We have B, and B. And only the naive, so I have to write, this is a row of naive B cells. There's only one of these Bs that actually has the BCR that recognizes the X antigen. So therefore, that's going to be attached to the receptor here, and we're going to get activation here with maybe a T helper cell nearby. And now we're just going to have proliferation. When we have proliferation, we're going to then have these B effectors. And then the effectors will become the plasma cells. They're also known as the plasma cells. And now they're displaying the receptors. Again, they're bound to that antigen X, the specific X receptor. And then once we get to the effectors, we could also have the memory B cells for the specific antigen. None of these other naive B cells are going to proliferate because we only need the specific one for X antigen. And then the effector action, we're going to then have the antibodies that are specific to antigen X made after this. So again, the memory, this is for encounter. And then that's going to be a strong secondary response. But then once we get down to the plasma cells or the B effectors, that's when we get tons of the X antibody. So this whole system will be proliferating. Okay, so B cell activation and T-dependent antigens. So again, we're going to get back here and roll back again. B cell activation plus the T-dependent antigen. So T. So that was like a generalization. The process here is a little bit of the nitty-gritty. So the BCR binds to the antigen. The antigen is moved inside and degraded into peptide fragments. Next, the fragments are moved to the MHC class 2. So if we go back here, we also talked about MHC up here somewhere. The surface of another cell in the major histocompatibility complex. That was for BCRs and TCRs. Specifically, I think TCRs. This is where we're going to have that T-dependent antigen. And then when they're delivered here to the class 2 molecules, this is for inspection by T helper cells. This is called the presentation. They rely on that presentation, the T helpers. They need to be presented. The TCRs on the T cells need to be presented something. If binding does occur, if the T cell binds to one of these fragments, then the T cell will activate the B cell with cytokines. If not, well, this actually causes the clonal expansion, which we talked about here, up from this section. But if no binding between the T and the fragments on the class 2 MHC, this will be an anergic B cell. And that means it's just going to stand for tolerance of the antigen. That's only if there's no binding between the T and the displayed fragments. That's what that picture is saying. Okay, characteristics of the primary response. Again, primary response. This is going to be 10 to 14 days for antibody accumulation. Some B cells will become plasma cells, as seen in that diagram about the clonal expansion or clonal selection. And the plasma cells are going to make antibodies, but they're also short-lived. Plasma cells will do apoptosis after a few days. The activated B cells continue to multiply and differentiate as antigen is present. So the antibody titer steadily increases. As long as there's that antigen, we're going to be making the antibodies in the primary response. The secondary response, however, again, this is where the stronger response due to memory. This is from additional exposure, and that's going to create a much faster secondary response. And it's also more effective. The pathogens are eliminated even beforehand. And vaccination will be exploiting that secondary response. The memory B cells will be responsible. They have increased numbers during the secondary response. When activated, these extremely important cells will start becoming plasma, and then of course that makes antibodies, and then this is even faster than the primary. So again, memory B cells, they remember, so then when they see that antigen again, they go through the process extremely fast. T-independent response with antigens in B cells in the humoral system will of course be the antigens with evenly spaced subunits or epitopes. For the B cells to recognize using their receptors. Again, T-independent, no T helper here. And lipopolysaccharide, as described earlier, from the outer membrane of gram-negative. This is a great T-independent antigen. Alright, that's going to be everything for the humoral side of adaptive immunity, and we'll get to the cell-mediated in a bit. So the humoral, again, uses the antibodies, antigens as specific BCRs and TCRs, depending on the antigen. If it's T-dependent or T-independent, then we're going to get the differentiation only for the specific naive B cell that has that receptor that is marked for that antigen. Then it's going to go to an effector if it needs the help of a T helper cell. It's going to go to the effectors, which is a plasma cell, and those are going to make tons of antibodies. And then afterwards, we get the secondary response where it becomes a memory B cell when it re-encounters it for a much faster process of creating antibodies. And vaccination is that secondary response in the humoral side of adaptive immunity. Thanks so much. Please stay tuned for the next part and do something nice for someone.